Hello viewers, today we are going to discuss about biological functions and disorders of insulin. Now we'll see the introduction of insulin. Insulin is a protein hormone which is secreted by beta cells of islets of Langerhans in pancreas. It plays an important role in metabolism, causing increased carbohydrate metabolism, glycogenesis, glycogen storage, fatty acid synthesis, triglyceride storage, amino acid uptake and protein synthesis. Thus, insulin is an important anabolic hormone which acts on variety of tissues. Major target tissues of insulin are muscles, liver, adipose tissue and heart. And it is essential for energy utilization and maintaining metabolic balance. Structure of insulin. Insulin is a heterodimeric protein, has been isolated from pancreas and it is crystalline in form. For crystallization, it requires zinc 2 plus ions. Zinc is also a constituent of stored insulin and normal pancreatic tissue is relatively rich in zinc. Insulin molecule is composed of two polypeptide chains called A chain and B chain, consist of 51 amino acids. A chain consists of 21 amino acids and B chain consists of 30 amino acids. In A chain, N-terminal amino acid is glycine and C-terminal amino acid is asparagine. In B chain, N-terminal amino acid is phenylalanine and C-terminal is threonine. Disulfide bridges in insulin. In insulin, both the chains are held together by two disulfide linkages. Cysteine 7 and cysteine 20 of A chain are joined to cysteine 7 and cysteine 19 of B chain respectively. In addition, the A chain carries an intra-chain disulfide linkage between cysteine 6 and cysteine 11. The importance of disulfide bridges when breaking of the disulfide bonds with alkali or reducing agents, it inactivates the insulin. And also, digestion of protein with proteolytic enzymes also inactivates the hormone. Molecular weight of insulin is 5734. Biosynthesis of insulin. Insulin biosynthesis begins with the translation of mRNA into pre-proinsulin, which is then processed in the endoplasmic reticulum and Golgi complex to form the active insulin hormone consisting of A and B chains, and C peptide is cleaved off. In the first step, gene transcription, that is, the insulin gene located on chromosome 11 is transcribed into messenger RNA. Then the messenger RNA exits the nucleus and moves to the rough endoplasmic reticulum. Then ribosomes on the rough endoplasmic reticulum translates the mRNA to produce pre-proinsulin, which consists of signal peptide A chain, B chain and C peptide. Pre-proinsulin is an inactive precursor. Then conversion of proinsulin. The signal peptide is cleaved off in the endoplasmic reticulum, forming proinsulin. Proinsulin folds properly assisted by chaperone proteins. Then proinsulin is transported to the Golgi apparatus where it is packaged into secretory granules. Inside these granules, specific enzymes such as prochrome convertase cleave the C peptide, resulting in mature insulin, which is composed of A chain, B chain and linked by disulfide bonds. Then insulin is stored in secretory vesicles within the beta cells. Upon glucose stimulation, insulin is released into the bloodstream through exocytosis. Now, moving into biological functions of insulin. In carbohydrate metabolism, insulin increases glucose uptake from extracellular fluids by the various tissues like muscles, adipose tissue, mammary glands, etc. Increases utilization of glucose for providing energy, which takes place in muscle, liver and many other tissues. Insulin enhances glycolysis because it induces the synthesis of key enzyme phosphofructokinase and also pyruvate kinase. And insulin increases aerobic oxidative decarboxylation of pyruvate to acetyl-CoA because it causes dephosphorylation of pyruvate dehydrogenase complex, which is thus converted to active form. Insulin stimulates glycogenesis in the liver and muscles by increasing dephosphorylation of the key and rate limiting enzyme, that is glycogen synthase, thus converting to its active form, and also stimulates the protein phosphatase 1 directly, which brings about dephosphorylation. Insulin reduces gluconeogenesis by repressing the synthesis of key rate limiting enzyme phosphoenol pyruvate carboxykinase by decreasing the transcription rate of the gene, and it also inhibits allosterically fructose 1,6 biphosphatase, another key enzyme for gluconeogenesis. Insulin dephosphorylates fructose 2,6 biphosphatase so that it is converted to inactive form, which increases the concentration of fructose 2,6 biphosphate in the cell, which in turn allosterically inhibit fructose 1,6 biphosphatase. Insulin action on lipid metabolism. Insulin decreases lipolysis in adipose tissue cells and consequently lowers plasma-free fatty acids. Insulin activates phosphoprotein phosphatase, which dephosphorylates the triacylglycerol lipase and thus converted into inactive form. At the same time, insulin activates phosphodiesterase, which degrades cyclic AMP and prevents phosphorylation and reactivation of triacylglycerol lipase. Insulin increases the extramitochondrial de novo fatty acid synthesis by making available of more substrate acetyl-CoA and also increasing the activity of acetyl-CoA carboxylase. Insulin induces the synthesis of ATP citrate lyase to increase cleavage of citrate so that more acetyl-CoA is available in cytosol. It induces the synthesis of acetyl-CoA carboxylase and fatty acid synthase, the cytosolic enzymes required for fatty acid synthesis. Insulin activates acetyl-CoA carboxylase by dephosphorylation of the enzyme, and it provides more NADPH for the reductive steps in fatty acid synthesis by stimulating HMP shunt pathway. Insulin action on protein metabolism. Insulin increases amino acids uptake by the tissues by enhancing the rate of synthesis of membrane transporters for amino acids. Insulin helps build proteins by increasing the amount of amino acids in cells. It also influences gene activities, controls the production of specific mRNA, and improves protein making at the ribosomes. Ribosomal translation is regulated in two ways. Insulin increases the production of polyamines, which are needed for making ribosomal RNA by boosting the key enzyme ornithine decarboxylase. Insulin also affects ribosome activity by adding phosphate groups to the 6S ribosome, a part of the 40S subunit. Insulin action on mineral metabolism and growth. Insulin lowers the level of potassium and inorganic phosphate in the blood by increasing glycogen production and glucose phosphorylation. Insulin promotes growth in living organisms and helps cells multiply in lab studies. Research using cultured fibroblast cells shows that insulin enhances the effect of growth factors like fibroblast growth factor, platelet-derived growth factor, and epidermal growth factor. Disorders related to insulin involve problems with its production, action, or both, leading to various metabolic issues including diabetes and insulin resistance, which can cause high blood sugar levels. A group of metabolic disease characterized by high blood sugar level due to either insufficient insulin production or the body inability to use insulin properly. Type 1 diabetes is an autoimmune disease where the body immune system attacks and destroys insulin producing cells in the pancreas. Type 2 diabetes is characterized by insulin resistance where cells do not respond normally to insulin and often accompanied by insufficient insulin production. Gestational diabetes develops during pregnancy and usually resolves after childbirth, but increases the rate of developing type 2 diabetes later in life. Insulin resistance is a condition where cells to fail respond normally to insulin, leading to elevated blood sugar levels. Pre-diabetes, a stage before type 2 diabetes where blood sugar levels are higher than normal but not yet high enough for a diabetes diagnosis. Hypoglycemia, low blood sugar, which can occur as a side effect of insulin treatment for diabetes. Insulinoma, a rare tumor of the pancreas that produces excessive amount of insulin leading to hypoglycemia. Other associated conditions such as polycystic ovary syndrome, it is an hormonal disorder that can cause irregular periods, excess androgen levels and insulin resistance. Metabolic syndrome is a cluster of conditions including high blood pressure, high blood sugar, excess body fat around the waist, and abnormal cholesterol or triglyceride levels that increases the risk of heart disease, stroke, and type 2 diabetes. Then type 1A insulin resistance syndrome is a rare genetic disorder characterized by severe insulin resistance, abnormal glucose homeostasis, ovarian hyperandrogenism, etc. Type B insulin resistance, it is characterized by severe impairment of insulin action triggered by the presence of insulin receptor antibodies. Thank you for watching the video. Have a good day.